In this video, proof of concept is shown for a lensless on-chip imaging termed LUCAS. LUCAS stands for Lensless Ultra-Wide Field of View Cell Monitoring Array Platform based on shadow imaging. Unlike conventional imaging setups, the LUCAS platform does not require any microscope objectives or other bulky optical components. For LUCAS, the specimen of interest is positioned on a microscope slide and is gently sandwiched by an identical cover glass. Using a vacuum pen, the sample is loaded onto the active region of the optoelectronic sensor array. The imaging platform then records the shadows, or lensless digital holograms, of each cell of interest within its field of view. Automated digital processing of these cell shadows can determine the type, the count, and the relative positions of cells within the solution. The field of view can span as large as 18 centimeters squared, with a depth of field of about 5 millimeters. The acquired diffraction images are then digitally processed by a custom-developed decision algorithm to enable both characterization and identification of target cells or particles. LUCAS offers a miniaturized platform that reduces the cost and significantly increases the throughput for imaging-based experiments, making it compatible with advanced state-of-the-art technologies in microfluidics and biological research.
Hi, I am Onur Mudanyali from Bio and Nanophotonics Laboratory in the Electrical Engineering Department of University of California, Los Angeles.
And I'm Anthony Erlinger, also from the Nano and Biophotonics Lab.
Today we will show you a procedure for high-throughput and lens-free imaging of a heterogeneous cell solution such as whole blood sample.
This procedure in our laboratory utilizes a novel imaging technique which we call Lensless Ultra-Wide Field Cell Array Monitoring Platform based on shadow imaging, which we call LUCAS for short.